All right, hello again, and today we're going to be talking about DNA and RNA. So you may be wondering, why does Mr. Seggerman have a map of the major naval encounters of World War II in the Pacific Ocean Fleet when I'm talking about DNA and RNA? Well, you guys did an activity with coding for the order of 20 fruits in a random order. And this is, you might not realize it, but not too different from that. Um, all of these attacks during the World War II on the Pacific Ocean, actually, in all of our Navy, so thousands and thousands of ships in all these different offensives were coordinated using not 4 letters like you had, but only 2. So you may recognize this. This is Morse code. It consists of 2 letters, which would be the dots and the dashes. So you can combine those in, several combinations here in order to make the letters that you need to spell out something. So imagine trying to coordinate all of this stuff that's going on, and think of how complex a war has to be, and using only 2 letters to do so. And hopefully you realize that your assignment of putting 20 fruits in order with 4 letters was not that difficult after all. The reason why I had you do that is because your strategic plan or the instructions for making a person is not too different from the activity that you did. If we look here, this is, this is obviously a computer-based image here, but there's all these letters. Your DNA is spelled out pretty much the same way using only 4 letters. We know that DNA ultimately codes for making proteins, and we're going to see how that works late in another lecture video. But this is done using just 4 letters, A's, T's, C's, and G's. Here's a diagram. This is actually a sequence from somebody's DNA, where you can see there's all these A's, T's, C's, and G's put here. And believe it or not, those 4 letters can code for every single protein that needs to be made to determine your traits. Remember, we learned that proteins are responsible for controlling chemical reactions in cell processes, as well as various other functions in the body for like structural components as well. And so proteins really are what determine most of your traits, and DNA codes for all of that, and just using 4 letters. Just to remind you about nucleic acids, this is what DNA looks like. DNA is just one of the nucleic acids that we'll look at. A nucleic acid is a polymer. So remember, poly means many, and so when we say it's a polymer, we mean it's got many of these smaller units. So if we look here at this molecule, here's a smaller unit, and there's another one attached to it, and another one attached to it. And so as you put all those together, we make this really long molecule called a polymer. The monomer of a nucleic acid, so this little guy right here, is called a nucleotide. So that should just all be review. If we look at the 3 parts of a nucleotide, again, you saw this briefly. We have a phosphate and a sugar. Those make up the backbone of our DNA molecule. And then we have this thing here called a base. I'm going to add something to this. I'm going to say it's a nitrogen base. I'm sure you're all smart enough you can figure out why it's called a nitrogen base, but in case you're wondering, it's because it contains nitrogen. So this part right here is the nitrogen base. And that is where our letters, our A's, our T's, our C's, and our G's are at. So this nitrogen base, the order of these bases, when we put our nucleotides together, is what determines your traits. So here are the 4 nitrogenous or nitrogen bases of DNA. We have thymine, which we abbreviate T, cytosine, which is abbreviated C, guanine with a G, and adenine with an A. You'll notice that guanine and adenine look very similar in their structure. Those are called the purines. You don't necessarily need to know that. Thymine and cytosine also look very similar. Those are the pyrimidines. But notice in all of these, there's at least one, or in case of the pyrimidines, there's 2 of them. Sorry, 3. 3 in cytosine anyways. And in the purines, there's also these nitrogens. So that's why we call them nitrogenous bases. So when we look at DNA, DNA does something called base pairing. So those base, we're referring to the nitrogen bases, and that's just one part of our nucleotide. Remember, if we draw our nucleotide, we've got the phosphorus here, we've got the sugar here, and then sticking off the sugar is this nitrogen base. So these nitrogen bases will pair up with another nitrogen base across from it on another nucleotide. So if we look here, cytosine bonds with guanine. And the way that it does that is through these things right here called hydrogen bonds. That should be a review from the biochem unit. We talked about hydrogen bonds. So it's just it's kind of a weak bond here that holds this guanine to this cytosine. So cytosine and guanine always bond with each other. If we look over here at this strand of DNA, you can see on this strand of DNA, so the one that goes up and then down and then up, here we have a C. Across from it on the other strand of DNA is a G. So cytosine is always paired with a guanine. Likewise, thymine and adenine always bond together. And you can see here there's the hydrogen bonds that hold them together. Now the reason why cytosine and guanine only bond with each other is because if you look, there's 3 spots for them to make a hydrogen bond. Whereas thymine and adenine, there's only 2 spots. So it's not possible for adenine to bond to something like a cytosine. It just doesn't work. So again, over here you can see everywhere there's a thymine, across from it's an adenine and vice versa. So if we look at the shape of DNA, this is a really recent discovery, or a fairly recent discovery. It wasn't discovered until about the 1950s. Until that time, we didn't know that DNA was what was, our molecule of inheritance. We didn't know that was responsible for heredity. But these two guys, this is James Watson right here and Francis Crick. They discovered the shape of DNA along with somebody called Rosalind Franklin. She was responsible for doing a lot of the imaging using X-rays that allowed these two to interpret it and then discover the shape of DNA. They won the Nobel Prize in chemistry in the 50s for discovering the shape of DNA. And Rosalind Franklin kind of got gypped, but she was later posthumously awarded the Nobel Prize for her work that contributed to that. So the shape of DNA, right here, you can notice it's kind of this corkscrew shape. It's called a helix. So it's this kind of winding staircase pattern. And because there's 2 strands, we call this a double helix. That's very important. DNA is a double helix. So you'll notice this kind of backbone strand that I'm outlining right here. Excuse me, that's going to be our phosphorus and our sugars. So in each nucleotide, the phosphorus and the sugars, they make up kind of the backbone, the edge of this. If you imagine taking a ladder and twisting it, so grabbing the top end and the bottom end and twisting the ladder, assuming it wouldn't snap, that's kind of what you get. These rungs then, or these steps in between them that hold the 2 sides together, those are our nitrogen bases. So you can imagine here is, if we're looking at, let's say, a ladder, so here's my ladder, kind of a wobbly ladder. We can compare this to our nucleotide. So our phosphorus and our sugar would be right there. They make up the rails. And then the nitrogen base would be the rung. And then over here, that'd be hydrogen bonded to another nitrogen base, which is connected to its phosphorus and its sugar. And so you'd repeat this the whole way down. So the phosphorus and the sugar hold the DNA molecule together on the side, and then the nitrogen bases, they hold the 2 sides together. So this is a double helix. That's the shape of DNA. So when we look at DNA in the cell, your cells have a lot of DNA. And to give you kind of perspective, this is an E. coli bacteria. That stands for Escherichia coli, but you just know it as E. coli. This is a teeny, teeny, tiny bacteria. I mean, they're so tiny that you can't see them unless you use a powerful microscope. And even then, the microscopes we have in class, they just appear like little specks on the slide. E. coli has 1 chromosome, and it's 4.6 million base pairs long. So that MBP stands for million base pairs. What that means is, if we look here, this is 1 nucleotide, and here's its base pair. So this together is 1 base pair. Imagine having 4.6 million of these in a chain together. Think of how big of a molecule that is. And that's the chromosome for E. coli. Now, E. coli only has 1 chromosome, whereas humans, remember we said, have 46. And we looked at one of our smaller chromosomes, the Y, which only men have Y. It has, remember, about 50 million base pairs. So one of our 46 chromosomes, and if you're a male, you have this Y, and one of the smallest chromosomes is huge, over 10 times the size of the one chromosome that E. coli has. So think about how much DNA humans have. This is the E. coli right here, and you notice all these squiggles around it. That is its DNA. What we've done is we've actually lysed. Remember, lyse means to cut. So we split open the cell, and all of its DNA is kind of spewing out. Well, E. coli's 4.6 million base pairs is about 1.6 millimeters in length. Now that's pretty small, but compared to this E. coli, that's 1,000 times its length. Because E. coli is absolutely tiny, and so this strand of DNA that's over 1,000 times the length of the E. coli can all fit inside and still have room for all the other cell parts. The way that it does that is by coiling around these things called histones. So that's a word you need to know. A histone is a protein. They're represented by these little orange balls in this picture. It's a protein that kind of acts like a spool. So if DNA is the thread, it would coil around the histones, which are the spools. And so as the DNA coils on itself and then coils around the histones, and then the histones coil around each other, and those coils of histones, which are coils of DNA, coil on top of themselves, and they keep coiling and coiling until you have coils of coils on top of other coils of coils, and then eventually you get a chromosome. So we've talked about chromosomes already, but now you just kind of maybe can appreciate just how complex that is. It's a chromosome is this nice, tightly packaged strand of DNA that's been coiled over on itself many, many thousands or millions of times in order to condense small enough to fit inside of the cell. So histones are the proteins that help DNA coil up and eventually make chromosomes. We know that DNA has to be copied. Think about the S phase and interphase when we talked about mitosis. DNA has to be copied in order for cells to reproduce. And so that process of copying our DNA is called replication. It's a fairly straightforward process. There are two enzymes that are involved. Remember we talked about enzymes usually end in the suffix ase. So there's a DNA helicase, that is an enzyme. Its main job is to uncoil the DNA. So remember, DNA, it's a double strand, and it's twisted around. And so this helicase uncoils it, and then it also splits it. So imagine it's kind of like a zipper. It uncoils the strand, and then it splits apart the two sides. The other enzyme that we have is called DNA polymerase. So let's think about this again. ACE means it's an enzyme, so it speeds up a chemical reaction. Polymerase, that probably means that it's an enzyme that speeds up the process of making a polymer. We know that DNA is a polymer, so DNA polymerase is the enzyme that makes that process go faster. The way that it works is it reads the original DNA strand. So helicase splits the two strands apart, polymerase reads each of those two strands, and then it assembles, this is really important, a complementary strand. When we say complementary or something complements something, it means it matches up to. And when we think about that, complementary strands, think of the base pairs. What's complementary to A? T. What's complementary to C? Well, that'd be G. So it's going to assemble a complementary strand using extra bases that are just kind of floating around in the cell. The other thing that it does is it proofreads the new DNA strand to make sure that there's no problems, no errors. All right, so what does this look like? Well, let me change color really quickly so you can actually see what I'm doing here. We'll use red. So here we have helicase. You'll notice it's kind of this corkscrew shape. It's going along here and it's unwinding our DNA, and it's also splitting it into its two strands. So here's a strand, here's the other strand. These little green circles on here, those represent the different bases. So they could be A's, T's, C's, G's. The DNA polymerase, and there's a couple different kinds of polymerase. I just want you to know that DNA polymerase does this job. So don't worry about 1 and 2 and 3. They don't have 2 shown on here, but what DNA polymerase does is it reads along what's called this template strand or the original strand, and it assembles a complementary strand. So this isn't actually copying this strand, it's actually making a complementary strand to it. So it's base pairing it, because if up here on this strand we have an A, this strand is going to be a T. If this is a G, right here will be a C. Now you may be like, well, why is it assembling a complementary strand and not an exact copy? Well, because base pairing doesn't work that way. It does this by pairing these up. But the good thing is, this strand, this new strand that's right here, will be exactly the same as this bottom template strand. This new strand right here, that's being base paired to the bottom strand, will be the exact same as this strand. So as both strands of DNA get read, so one here, and one here, the new strands, this red stuff that comes out, will then join together and make a complete new strand of DNA. This process actually, you can actually see this happening with certain microscopes. So if we use an electron micrograph, which is a really, really powerful microscope, it uses electrons to make the image. Here you can actually see this happening. So if I use my pen here, there'd be a DNA helicase right here that would be splitting apart the two strands. And there would be a DNA polymerase reading this strand, and one reading this strand. And they would be assembling a new strand of DNA. One would come off here, and one would come out here. And then eventually, this one and this one will join up to make a new strand of DNA. And so on the back side of this, we call this a reading frame, by the way, this area that's open. On the back side, there's another helicase that kind of re-zips it. And this whole thing is going to move along the DNA strand. So this helicase right here will unzip it. This one comes along behind and re-zips it. Imagine like on a tent. Tents usually have 2 zippers. If you zip one, it opens the tent flap, and you follow it with the other one, and it closes it. That's kind of what's happening here. All right, so our other nucleic acid we're going to look at is RNA. RNA is very similar to DNA. It's just different in a couple key ways. One is that the sugar in RNA is ribose sugar. I don't think I mentioned it, but in DNA, the D stands for deoxyribose. It's just a kind of sugar. In RNA, the R stands for ribose sugar. The N you already know is nucleic and A is acid. So DNA means deoxyribonucleic acid, whereas RNA is ribonucleic acid, and that has to do with the sugar. RNA is single-stranded, so if we look over here, here's DNA, here's RNA, double strand, single strand. RNA, it's best to think of as short copies of the DNA. Our DNA has to stay inside of our nucleus. It's the master blueprint, but the RNA is short little copies. So imagine like, well, let's use the blueprint example. If you had a blueprint for making a big, huge building, like let's say the First National Bank building downtown, and you only had one copy of it, you wouldn't want to give that to just any old worker to go, put up an I-beam or something. You could give them a copy of part of it that they would need. And so that's how the RNA is. It's a short copy of the instructions that can be used for a specific task. It's not the entire thing, it's just a short copy of it. The important thing about that is RNA can leave the nucleus. Our DNA, it is stuck in the nucleus. It can't leave. There's 3 types of RNA, which we'll talk more about in the next lecture, but there's messenger RNA, which is oftentimes called mRNA. And then there's ribosomal RNA. It's part of the ribosomes. It's called rRNA. And then transfer RNA. Its name kind of gives it away. It moves things around. It's called tRNA. Again, we'll talk more about those later. One key difference, and this is RNA again, and so I guess it says it right here. Notice we have adenine, we have guanine, we have cytosine, but RNA has this thing called uracil. Notice there's no thymine. In RNA, instead of thymine, we have uracil. So U instead of having a T. So just to kind of summarize things up, if DNA replication is where we make a copy of DNA, transcription is how we make RNA. There's one enzyme that's used here. It's called RNA polymerase. Again, that's kind of gives away what it does. ACE means it's an enzyme. It speeds up reactions. Polymer means we're making polymers. That's the reaction that happens. And the polymer we're making is RNA. So pretty self-explanatory. What RNA polymerase does is it unwinds the DNA and then it assembles RNA again by doing this base pairing. But here's one little nuance. Because these are our DNA base pairs, when we base pair, we don't have any T's in RNA. So any place we have an A in the DNA, we base pair that to a U in the RNA. Otherwise, everything else is the same. A T in the DNA gives us an A on the RNA. So if we look here, here's this polymerase enzyme. It's reading our DNA, and it's assembling these base pairs. So notice every time there's an A on the DNA, it puts a U in the RNA. And there you have it. That's it.